Bioluminescent proteins, nature's glow sticks for visualizing biology. Bioluminescent proteins, nature's glow sticks for visualizing biology. Biological organisms have always fascinated scientists with their diverse and extraordinary abilities. One astonishing phenomena found in certain organisms is the production of light, known as bioluminescence. In essence, bioluminescence is the process in which organisms emit light through a biochemical reaction. It occurs across a wide range of organisms including bacteria, fungi, algae, jellyfish, and fireflies. The light emitted can be of various colors, including green, blue, red, and even bioluminescent flashes. One of the most intriguing aspects of bioluminescence is that it occurs spontaneously without the need for external light sources. Organisms capable of bioluminescence produce this natural glow through the action of specific proteins called bioluminescent proteins. Bioluminescent proteins are responsible for the emission of light in bioluminescent organisms. They are often found in conjunction with other proteins and chemicals that participate in the light prod. Using reaction. One of the most well-known bioluminescent proteins is green fluorescent protein, GFP, discovered in the jellyfish Equoria victoria. GFP has revolutionized biological research as a powerful imaging tool due to its ability to emit a vibrant green light when exposed to ultraviolet or blue light. The discovery of GFP sparked a scientific revolution in the field of biology. Scientists quickly recognized its potential as a molecular marker for visualizing and tracking specific proteins within live cells and organisms. By genetically fusing the gene encoding GFP with a gene of interest, researchers can produce a hybrid protein that fluoresces under specific conditions. This technique, known as GFP tagging, allows scientists to track the movement and localization of specific proteins in real time, providing invaluable insights into their function. The use of bioluminescent proteins extends beyond mere visualization of cellular structures. Researchers have harnessed their power to study a myriad of biological processes. For example, by fusing bioluminescent proteins with enzymes involved in metabolic pathways, scientists can monitor the activity of these enzymes in living organisms. This approach has deepened our understanding of various diseases, such as cancer, and has facilitated the development of drugs that specifically target cancerous cells. Moreover, bioluminescent proteins have also been utilized to study the dynamics of gene expression. By fusing them with regulatory regions of target genes, scientists can track the activation and repression of genes in response to environmental cues or physiological changes. This enables researchers to delve into fundamental questions about development, differentiation, and disease progression. In addition to their applications in basic research, bioluminescent proteins have proven to be invaluable tools in clinical applications. One such example is in the field of molecular imaging, where bioluminescent proteins are used to track and visualize tumor growth and metastasis. By engineering cancer cells to express bioluminescent proteins, scientists can monitor the progression of the disease and evaluate the efficacy of potential therapies. This non-invasive imaging technique has the potential to revolutionize cancer diagnosis and treatment by enabling real-time monitoring of tumor response to treatment. The versatility of bioluminescent proteins extends even further with the discovery and engineering of new variants that emit different colors of light. Despite the significant contributions of bioluminescent proteins to scientific research, there are still challenges to overcome. One challenge is the optimization of brightness and stability of these proteins in various experimental conditions. Researchers are actively working to engineer new variants with improved properties, such as increased brightness and resistance to degradation. Additionally, advancements in imaging technologies and techniques are required to better exploit the full potential of these proteins. Bioluminescent proteins have opened up new avenues for studying biological phenomena, allowing scientists to explore intricate processes that were previously hidden from view. Their ability to emit light naturally, without the need for external sources, has made them invaluable tools in the field of molecular imaging. One area where bioluminescent proteins have made significant contributions is in the study of cancer. By engineering cancer cells to express these proteins, researchers can monitor tumor growth and metastasis in real time. This provides valuable insights into the effectiveness of different therapies and allows for the development of personalized treatment strategies. Furthermore, the non-invasive nature of bioluminescent protein imaging minimizes patient discomfort and reduces the need for invasive procedures. In addition to studying cancer, bioluminescent proteins have been harnessed to investigate gene expression dynamics. By fusing these proteins with regulatory regions of target genes, scientists can track the activation and repression of genes in response to various stimuli. This has shed light on the complex processes underlying development, differentiation, and disease progression. The discovery and engineering of new variants of bioluminescent proteins that emit different colors of light have further expanded the capabilities of these tools. Researchers can now simultaneously track multiple proteins or pathways within the same cell or organism, providing a more comprehensive view of biological systems. This multiplexing capability has significantly enhanced our understanding of the intricate interactions within cells and has the potential to uncover new insights into cellular functions. Despite the progress made, there are still challenges to overcome in the field of bioluminescent protein research. One such challenge is optimizing the brightness and stability of these proteins under different experimental conditions. Researchers are actively working to engineer variants with improved properties, such as higher brightness and increased resistance to degradation. Furthermore, advancements in imaging technologies and techniques will be crucial in fully harnessing the potential of bioluminescent proteins. Bioluminescent proteins are truly remarkable tools that have revolutionized the field of biology. These proteins enable scientists to visualize and understand complex biological processes in ways that were previously unimaginable. Bioluminescence, the ability of certain organisms to produce light, has fascinated researchers for centuries. Through the study of bioluminescent proteins, scientists have gained valuable insights into various biological phenomena. One of the most well-known bioluminescent proteins is green fluorescent protein, GFP, which was discovered in the jellyfish Euphoria victoria. GFP emits a vibrant green light when exposed to ultraviolet or blue light, making it an excellent tool for visualization. By genetically fusing GFP with a gene of interest, scientists can track the movement and localization of specific proteins in real time. This technique, known as GFP tagging, has revolutionized the study of protein dynamics, and has provided valuable information about protein function. Bioluminescent proteins have not only enhanced our understanding of cellular structures and protein dynamics, but they have also been used to study metabolic pathways and gene expression. By fusing Bioluminescent proteins with enzymes involved in metabolic pathways, scientists can monitor the activity of these enzymes in living organisms. This approach has provided insights into various diseases, including cancer, and has facilitated the development of targeted therapies. Furthermore, the fusion of bioluminescent proteins with regulatory regions of target genes allows researchers to study the dynamics of gene expression. This has provided valuable information about regulation of genes in response to environmental cues or physiological changes. By tracking gene activation and repression, scientists can gain a deeper understanding of development, differentiatio, and disease progression. The applications of bioluminescent proteins extend beyond basic research and have significant implications in clinical settings. In the field of molecular imaging, bioluminescent proteins are used to track and visualize tumor growth and metastasis. By engineering cancer cells to express these proteins, scientists can